To add another layer to the confusion around the genetic origins of obesity, we're going to, in this unit, explore the concept of epigenetics and obesity. So what is epigenetics? So we used to think that basically our DNA pattern, our genome, affected risk of disease based on what kind of genes were or weren't expressed based on that particular DNA sequence, okay, that we find in our genes. However, epigenetics isn't about changes, it's not about signal nucleotide polymorphisms, it's not about mutations to genes. The genes are staying the same, the nucleotide sequence is staying the same. So what epigenetic changes do is it doesn't change the DNA sequence. What it changes, though, is whether a particular gene product is actually expressed or not. Okay, whether, for instance, that region of the gene can be read and coded into a protein. And for instance, if all that DNA is wrapped tightly around a histone, it's harder to read it and express the gene product. Or if there's tags, these methyl group tags on that DNA sequence, then again, it's harder to read and harder to express that particular gene, okay? So these epigenetic changes seem to affect gene expression more than the genetic sequence, or instead of the genetic sequence, okay? And what's really interesting about epigenetic changes is that they seem to be passed down from generations. Like there are things that, for instance, your grandparents might have experienced in their life that is recorded in their DNA via these epigenetic tags. And you could inherit those epigenetic tags that then affect not your genetic sequence, but whether your genes are actually, like I said, expressed, okay? So be careful, because this is kind of a big area with a lot of question marks. There is still a lot to be known, so we don't want to assume too much, because this is still, we know that it exists, but we're still exploring this concept more and more, okay? So like I said, these epigenetic modifications record what a cell experiences on that genome. Okay, and the three main epigenetic tags that we know of, that there's research to support, are something called DNA methylation, histone modification, and non-coding DNA. Although most studies into epigenetic modifications focus on DNA methylation, which is what we'll focus on as well. Like I said, these tags, they can just basically alter gene expression. And like I said, what your mother experienced, what you experienced, perhaps what your grandfather, your grandmother experienced, maybe further generations as well, that is recorded, okay? And these external factors, like a person's diet, like whether they smoked, like whether your mother had anxiety when you were in the womb, these kind of things are recorded kind of on top of our genome, and again affects gene expression. And we do believe that is one of the things that links genetics and epigenetics to diseases like obesity and cardiovascular disease and a whole slew of other ones. Okay, so I want you to watch this video that explains it maybe a little bit better than I just did, but also to just give you a little bit more of kind of visual clarity about what I'm talking about. When I'm talking about histone modification, this is a histone right here, that brownish blob there that the DNA is wrapped around. And the tighter the DNA is wrapped around that histone, the harder it is going to be to read. Okay, so that's one of the main epigenetic changes. But the main one that a lot of research focuses on are these methyl groups that can be kind of added on to certain parts of the DNA sequence, specifically the cytosine guanine sites of certain genes. Okay, and then another epigenetic tag that we know less about, and there's less research, especially around obesity with this, is this concept of microRNAs or non-coding RNAs, which is considered part of the epigenome as well. Okay, so I've already kind of explained these, but to go over that a little bit more, like I mentioned, though, that methylation of our DNA, notice our DNA sequence, our double helix, is not changing. That's not what's changing. The nucleotide sequence is not changing, but these little tags around that DNA sequence are affecting whether genes are expressed or not. And they can basically silence transcription. Okay, and like we said, that's heritable. Okay, histone modification, we already talked about that. They're like spools that DNA can wind around, and modifications to those histones can again affect gene transcription and gene expression. Okay, and then like I said, these microRNAs, or sometimes called non-coding RNA, are mRNA that's produced from a gene that does not actually code for the gene product like it was supposed to. It's not actually translated into a protein, but again, that's not one of our main focuses for this unit. Okay, so what are some of the evidence pieces that suggest that there is an epigenetic basis for obesity? So one of the major ones is that we know that what a mother eats when she is pregnant and whether she gains a lot of weight when she's pregnant, that can affect her offspring's risk for obesity. Okay, and it's probably not just through epigenetic changes. Okay, but we believe that that is one of the main mechanisms by which a mother with obesity increases the risk of a child with obesity. Okay, and conversely, when mothers lose weight during pregnancy, if they were larger, that can actually decrease the risk of obesity in their offspring. Okay, remember when we learned about that fat mass associated gene, the FTO gene, that locus codes for a DNA methylase enzyme. So it might be that the change in expression of that gene is affecting methylation, and what makes that genetic change affect obesity is actually through epigenetic means. Okay. We also see changes in methylation when people and mice, well, more studies in mice, are fed a high-fat diet. We actually see a reduction in methylation of the MC4R gene, the melanocortin receptor, which is part of the appetite sensing pathway and the satiety pathway in the hypothalamus. Okay, like it says here, when pregnant rats were fed a high-fat diet, their offspring had an increased risk of obesity as well. So again, something's happening. We believe something's happening as far as what we do, what we expose ourselves to, what our mother exposes herself to, what our father, our grandparents expose themselves to. That's leaving like this mark on our DNA, which in turn is affecting our risk for a bunch of different things, including obesity. So one of the big studies that helps us understand the concept of how epigenetics can relate to, can affect phenotype basically, and in particular how it can affect obesity phenotype, are these agouti gene mouse studies, okay? And what basically happened, what you need to know to understand this study is, first of all, that agouti is a gene that codes for coat pigment, the coat pigment melanin in mice, okay? And we know that when that agouti gene of mice is unmethylated, okay, that mouse is more likely to have a yellow coat and it's more likely to be obese. Okay, unmethylated agouti gene, yellow phenotype, obese phenotype. Okay, all we did with these pregnant unmethylated agouti mice is we fed them either a methyl-rich diet, a diet that was rich in methyl groups, or kind of a standard diet. When those mice were fed, the pregnant mice were fed a standard diet, a normal diet, the offspring was just like the mother, probably because methylation didn't change. Okay, or that's what the conclusion of the study was. However, when we fed these pregnant agouti mice with the yellow obese phenotype a diet rich in methyl groups, remember, because the agouti gene is unmethylated, or more unmethylated in the mother here, their child had a completely different phenotype. The coat was brown, more brown, and the offspring were more lean as well. Okay. So to kind of show you the real results of that study as opposed to just the ones that are represented in that cartoon, is we see kind of the different in coat colors. Okay, so these yellow mice here, okay, which especially with this one you can tell is larger than this mouse here. Okay, it shows kind of the difference in methylation. Okay, as far as these mice go. So these yellow agouti mice are more likely to be unmethylated at the agouti gene. And when we get closer to this, what we call pseudo-agouti phenotype, here we have an increase in methylation. Okay, so if we look, so results from the same study, if I look at I'll call it 1 versus 2 here. So we're looking at CPG sites, those sites that are methylation prone. If we look at the unsupplemented, the ones not fed a methyl-rich diet, there is a smaller percent of cells that are methylated. That makes sense. Whereas if we supplemented that diet with methyl-rich groups, we see a much higher percentage of cells we see a lot more number of cells that had that methylated status. Okay, and again, the more highly methylated those sites were, the more likely the mice were to have that brown and lean phenotype. Again, the genome is the same, basically. Okay, the only difference are the methylation factor in these genes. Okay, which again suggests that there's something going on in the epigenome that's affecting perhaps gene transcription and the production or gene translation and the production of that gene product. And this slide just summarizes that same thing over again, but it kind of just shows in a different way. Again, we're more likely to see that thin, healthy brown appearance when we have more methylation. Okay, whereas we are more likely to have that obese and yellow phenotype when there's less methylation. Again, giving an argument for DNA methylation as being one of the epigenetic changes that can affect risk for disease, and in this case, obesity. And now would be a perfect time for me to play this interactive video for you guys, or to ask you guys to do it yourself. It's not online right now. It's supposed to be back online in spring 2021, but that's like right now. And I just checked and it's not. But maybe by the time you watch this, it is. And you want to check out this site because you're actually it actually allows you to move the dial. So you can increase histone so in this activity, what was really cool about it is you could play with histone modification and see how that ultimately relates to gene expression. And again, it just helps to maybe make sense of this concept of epigenetics affecting phenotype. Okay, but to kind of put this whole unit in perspective, to give you some take-home messages, we have learned that in some rare cases of obesity, the main driving force is some sort of change in a single gene, okay, or what we call monogenic causes of obesity. However, most cases of obesity that are genetically linked, or the genetics is a major driving factor, it's more of a polygenic situation where changes, the accumulation of changes in probably a lot of different genes comes together to increase propensity for obesity, probably through primarily changes in appetite regulation as far as the hypothalamic control of appetite regulation. Okay. In addition to changes to our genome that can increase our risk for obesity, there's also changes to the epigenome. Those tags on top of the genome are associated with the genome that can affect not the nucleotide sequence, but whether a gene is actually expressed as a protein. And what's cool about looking at the epigenetic origins of disease is that these are more modifiable. We can't change our genetics, but we can change our epigenome. How do we change our epigenome? By the things we expose ourselves to. And we're still trying to figure out what is the healthiest way to support a healthy epigenome. When it comes to obesity, the biggest kind of finding so far is that a high-fat diet can really alter the epigenome in a way that it promotes dysregulation of appetite, in a way that it promotes obesity. But there's probably a lot more to still learn about this subject. But again, it gives kind of more emphasis to why a healthy diet is so important and what we expose ourselves to is so important, because like that can leave tags on us that affect our behavior and our phenotype, and that we can also pass down to our children as well. So something to keep in mind. But also something to keep in mind about this unit is this concept of genetics loads the gun, but the environment pulls the trigger. Okay? There are some people that, yes, are what we might call genetically resistant to obesity. We know these people. It might be you. No judgments if that is you. I used to be very jealous of people that were perhaps genetically resistant to obesity, but you know, we all have our differences, and that makes us beautiful. But some people are genetically resistant in the sense that, you know, even if they're living in a very toxic obesogenic environment, they still don't eat a lot. Their appetite isn't strong. They're all the kind of energy balance associated factors are so promoting of a leaner phenotype that, you know, they're kind of resistant to a toxic environment. Okay. That said, there are some people that are genetically prone to obesity. A lot. Okay. We don't know the exact number, but a lot. And if those genetically prone individuals are in a healthy environment, well, they're more likely to show a phenotype like a genetically resistant individual. But right now, we live in a toxic, obesogenic environment where those that are genetically prone to obesity are more likely to develop it because, as we said earlier, the environment pulls the trigger. Okay? Genetics loads the gun, and what our environment kind of promotes in us pulls the trigger and promotes obesity. Okay, so we can't change genetics. We can alter epigenetics, but we're still kind of learning how to. But what we can really do is focus on regulating our environment and, you know, focus on what we can control, which might be more behavior oriented, which we'll talk about later on in the semester.